Good afternoon, everybody. It is my great pleasure to have a chance to discuss with you regarding the molecular detection of the SAR-CoV-2 for pandemic control of the COVID-19 through active case finding. My name is Dr. Vip Viprakasit. Actually, I'm a pediatric hematologist from Faculty of Medicine, Sirat Hospital, Mahidol University, Bangkok. You might be surprised that why I'm a pediatric hematologist I'm giving this lecture on this topic. Since the pandemic of the COVID-19 started in 2019 following with last year 2020, I have taken a very important part participating in the pandemic control in Thailand. Owing to the fact that I've been trained as a molecular biologist at the University of Oxford in UK for many years ago. So I used my knowledge to start the laboratory to helping on the molecular detection of the COVID-19 in Thailand. And this is the recent interview that I make for the Straits Times Singapore newspaper regarding the current situation of Thailand regarding lockdown and opening the country up for the tourists and the business because of the drive of the economy is very important. As a basic background to remind you regarding the differences between the COVID-19, which is meaning the corona viral disease 2019, which is caused by the virus, and we call this virus as the SAR-CoV2, or which stands for the severe acute respiratory syndrome coronavirus 2. So it started in December in 2019 at Wuhan, China. And after that, there's the increasing number of the spreading. Even now, as I'm talking here, the pandemic is not over yet. Detection of the SAR-CoV-2 viruses is very critical for the pandemic control for the COVID-19. The virus itself is the RNA virus, and this is a composition of the virion and the genome of the SAR-CoV-2. It's composed of the spike protein and also the membrane protein, the envelope protein, and the nucleocapsid or the N protein. Inside is the RNA of the virus, which is the blueprint of the viral copy to amplify itself in our human bodies. So detection of the virus can be done using the molecular technique in order to identify all these important viral genes. Of course, the SAR-CoV-2 is belong to the coronavirus families. So detection of the genetic variation at the SAR-CoV-2 make a detection of this virus easier compared to the other technique. Because of the COVID-19, the SAR-CoV-2 virus has a unique sequence that can make distinguish from the other coronavirus. So we use the molecular detection to identify all those genes, in particular the S gene and the N genes, and also some other gene which seem to be a kind of housekeeping that also similar to the other coronavirus, such as the RDRP or the ORF1AB genes. After the infection of the coronavirus of the SAR-CoV-2, we export to the virus, and during the first or up to 2 weeks, the virus go into your bodies. At that period, it's very unlikely to detect the virus itself. But after the incubation period, the virus will bind to the ACE2 receptor, especially in the nasopharyngeal region, and start to amplify, increase the copy itself. So at this period, we can identify by using the polymerase chain reaction or the PCR, that using this technology to identify the viral RNA. And after a period of 1 week, you start to see the antibody developing, especially the IgM and followed by the IgG. So detection of the viral RNA starting from the nasopharyngeal region and followed by the respiratory tract broncholavage to the stool PCR has become a cornerstone of the viral detection. And after 3 to 4 weeks, when the antibodies start to develop, the level of the virus will start to go down, and the detection by the molecular detection will become decreasing and later on to be negative. So we use this concept to identify the virus itself as the gold standard. Of course, you need to take the sample from the nasopharyngeal region by the nasal swab. And later on, there is also some developing the sample collection by the saliva, or you can do the nasal swab. But the best remains the nasopharyngeal swab that we can have the highest copy of the virus for detection. After we take the swab, the virus has to be kept in the VTM or the UTM until the testing, and we are extracting the virus out to purify the RNA and perform the reverse transcription to make this as a cDNA and identified by the classical PCR technology, which I'm going to explain in detail. And this cDNA, after this amplification, we can use it for the further identification of the variants, the strain, by using the next generation sequencing or direct sequencing of the virion of the viral genome itself. The process of the nasopharyngeal swab to obtain the sample is very critical. So you need to do a proper procedure to take the adequate virus and keep the virus in the viral transport media or the VTM and store it at 2 to 8 degrees up to 3 days before the RNA extraction. You have to deactivate the virus by some of the chemical inside the transport media or using the thermal deactivation, heated it up. And after that, we perform the RNA extraction. And usually this process of RNA purification takes around 30 minutes, 15 minutes, up to 40 minutes, 45 minutes. After we got these RNA, we reverse transcribe into the cDNA and we perform the polymerase chain reaction. This is just to remind you that in this process, we design the amplification primer to specifically bind with the RNA sequence. And using the polymerase chain reaction, the RNA polymerase, to amplify the target region of the genome of our interest. So in the different design from the different company or different optimization, we select different region of the SAR-CoV-2 viruses to be amplified. Usually, we need to use the specific region, especially the end genes of the virus, which is very important. And in Thailand, for detection of the virus, you have to have a positive end genes. So in this end gene, in different setup, have a different design for the forward and the reverse primer, as I show on this slide. Together with the end genes, we also amplify the other region of the virus itself as a control. For example, like ORF1AB genes, the RDRP region of the virus, and also some other genes such as the E genes or the S gene as well. So In Thailand, the detection of the SAR-CoV-2 by the RT-PCR need to have a positive of at least 2 genes. One of that has to be N genes. During this process of the viral detection, we make use of the technology called the real-time PCR to make the detection quicker and more rapid. So in this process of the RT-PCR, the real-time PCR make use the fluorescent probes that have been added into the PCR process. In this process, the fluorescent probe is quenched at the beginning of the PCR process. But after we amplify of the target viral DNA sequence, the probe will be dequenched and bind with the region of our interest, which is the viral genome, and start to release the fluorochrome out that can be detected by the fluorescence sensor at the PCR machine. So you will see the pickup of the fluorescent positive signal compare between the positive signal and the negative signal. And this fluorescent signal intensity and the level that we can start to detect by the different cycle will represent the different viral load. So if the CT or the copy per reaction is lower, meaning that the viral loading is higher for this detection. The process of the RT-PCR assay has to be done in a negative pressure room in order to contain the virus inside the room and take the air out to clean it before we release it into the outer environment. Especially in the process of the RNA extraction and combining the RNA extracted with the master mix of the PCR. After this process of RNA extraction, the virus will become deactivated. So we can do the process of the PCR and RT-PCR in the regular laboratory environment, as I show on this slide. The most important part of this molecular detection is the quality of the RNA. So if the RNA quality is not good enough, meaning that the integrity of the virus will be lost, so the detection will become lower. So I have designed this kind of the biosafety mobile unit, which is as the cabinet and also the mobile on a pickup truck for Thailand. And this system has been proven to be very effective. And now we have over 50 this mobile unit to be used for the active case finding for control the COVID-19 in Thailand. And in this picture, I'm very, very proud to present this project to His Majesty the King, and he is very supporting of this project for our country. This work made me become the Outstanding Researcher of the Year in 2021, and I also received the Prime Minister Award for Innovation for Crisis from the National Innovation Agency of Thailand. So during these 2 years of the SAR-CoV-2 pandemic, my company and my laboratory, which called AdGenes, has a chance to perform the RT-PCR testing, nearly 350,000 testing, around 20,000 last year from April to December. As you know that during last year, Thailand has become very successful to control the SAR-CoV-2 in Thailand, especially with the alpha strain. However, this year we have the spreading of the Delta strain, and this Delta strain started, as you can see on this slide, in April this year. You can see a very peak of the sample that we performed in our laboratory, and The highest is in July and August, June and July, August. You can see that the figure is very high. So the false positive rate is lower than 0.5%, and the false negative rate is lower than 1%. So the accuracy of the RT-PCR is 99%. Besides, we perform the conventional RT-PCR using the fluorescent probe and the real-time PCR, as I discussed earlier. Another technology that we have been experiencing, which is using the POC, or point of care molecular testing. So this point of care is much easier than the conventional RT-PCR. After we collect the sample in the spatial transport media, which is specifically designed for different POC machine, we loading the sample directly to the cassette and put the cassette, place the cartridge into the molecular testing machine and have it run. Actually, inside the machine also use the same technology of the RT-PCR that I described, but it just have less human handling the sample itself, and the process of the RNA extraction and amplification is occur simultaneously at the same process. So in the system that we use, besides the COVID-19 or the SAR-CoV2, we can also detect the other viruses, respiratory viruses, such as the other coronavirus or influenza, the parainfluenza, or the respiratory syncytial virus as well. So totally around 22 other different viruses and pathogens can be detected simultaneously using the POC machine. And the result will be available very quickly within 2 hours. So besides this POC, this RNA that after the extraction can be after detected by other molecular technique. And one of a very interesting and promising technology is by using the CRISPR technology to detect and identify the region of our interest. This cut a lot of hassle of the process of the PCR because this is an isothermal amplification. However, this isothermal amplification and detection with CRISPR or without CRISPR is not widely used in Thailand, and I think the reason for that is because the validity of the test itself require a further work on that, and also the process of this isothermal amplification still require the negative pressure room to perform the test itself as well. And finally, you can also use the digital droplet PCR to amplify and identify your viral DNA of your interest. Again, the limitation of this digital droplet PCR is the cost of the instrument itself. So this molecular detection is not widely used for the COVID-19 during this pandemic. We also have experience of applying our RT-PCR for the Phuket Sandbox program, because as you all know that tourist industry account for over 10% of Thailand GDP. So we take care of the swab center, especially at the Phuket International Airport, and 2 additional center in the Phuket City at the Central Forestier and also the King Power Shopping Mall. So this is our information about the Phuket Sandbox, that the people go through our system, do the registration, perform the test at the airport, and we perform the RT-PCR and send the sample. So the airport lounge turned into the COVID-19 laboratory. So far, we perform 32,000 testing since July the 1st until this is the collection data until September this year. Only 0.2% of them have been detected with the COVID-19. However, during this process, only 3 months, we generate the income by the tourists to Phuket, at least 1 billion baht, and that's a lot of money. Only 74 cases have been identified, but most of them are no symptom. 27 of them were mild and they no require ICU. So the Phuket Sandbox seemed to be a very successful program. And this is just to show you the different country that come with the different infection rate. So the pandemic has given people the opportunity to step up in the way that their typical job might not allow. And again, I think I really like this quote from this professor, because actually as a pediatric hematologist who has knowledge in molecular biology, I turned into the molecular biologist and helping my country to cope with the COVID-19. Thank you very much for your attention, and I will be very happy to take any question. Thank you.